The overall goal of this procedure is to demonstrate the use of functional near-infrared spectroscopy over the sensory and motor regions of the brain with simultaneous kinematic and EMG monitoring during different motor tasks. This is accomplished by first placing the fNIRS optodes onto the participant's head and aligning it with CZ and the pre-auricular points. The second step is to place the EMG electrodes over the muscle bellies of target muscles and the reflective markers on the anatomical landmarks. Next, collect data from the fNIRS, EMG, and the motion capture systems simultaneously while the participant is performing motor tasks. Ultimately, fNIRS is used to identify differences between the brain activation patterns in those with unilateral and bilateral childhood-onset brain injury and those who develop typically.
The main advantage of this technique over existing methods like fMRI is that this technique allows us to study the performance of upright and functional tasks such as gait.
This method can help answer key questions in the neurorehabilitation field, such as how does the brain reorganize after an injury, and what are the consequences of that injury on the performance of motor tasks.
The use of this technique in studying motor disorders allows us to measure brain activity while simultaneously quantifying intended as well as unintended movements through motion capture and electromyography.
Generally, individuals new to this method will struggle because accurate setup requires careful hair preparation and placement of fNIRS optodes for optimal signal quality.
Assisting us with the demonstration of the procedure today will be Dr. Diane Gallagher.